Hey guys, okay, my group is going to do our presentation on antimicrobial peptides. For the first thing off the bat is what are antimicrobial peptides? In simple terms, they are small proteins made of amino acids. They are made from cells, made by the immune cells like macrophages and neutrophils. They are also made by glands in your mouth. Different types are defensins, cathepsins, and histins. Locations in the body. Where are they found? The easy answer is anywhere germs can sneak in. They're in the skin, lungs, mouth, gut, intestines, body fluids, and immune cells. They're basically anywhere that can help and stop infections before they begin.
So now we're going to get into what they actually do. Well, they're the first line of defense when it comes to the rest of the immune system. They make infection harder for everyone because they stop bacteria from sticking to our cells, and they stop spreading and growing by causing the other bacteria to leak out and die. The main importance of the antimicrobial peptides is they keep the balance of good and bad throughout our body. They support the microbial homeostasis in the body, actually. So a big overview, and one of the main things that they do is they're like tiny crazy soldiers in your body. They poke holes in the germs and they happily watch them bleed them out. So your whole body is safe.
Understand the mechanisms of antimicrobial peptides, we first need to understand a little bit of the structure of bacteria. Most bacteria are composed of free-floating genetic material, including DNA and RNA, and ribosomes in the cytoplasm. Surrounding these important structures, bacteria have a cell membrane that helps control the inner and outer environment of the bacteria, along with a cell wall, which provides the cell with protection and structure. Through the mechanism that antimicrobial peptides have developed, we will be able to see how these little peptides become heroes by killing pathogenic organisms to keep us safe.
Antimicrobial peptides, otherwise known as AMPs, come from various platelets, epithelial, and immune cells anywhere in the body. After being made, AMPs circulate the body, inspecting for pathogenic bacteria. Once they detect the bacteria throughout the body, they fight the bacteria through various mechanisms and kill them, thus making AMPs a component of our first line of defense in the immune system. But how? One mechanism is through damaging cell membrane integrity. AMPs are amphipathic, meaning they have both hydrophilic and hydrophobic parts. This is what allows them to interact with the phospholipid bilayer of the cell membrane to create pores within the membrane. By squeezing enough amps into the membrane, a pore will be created, thus leading to the death of the cell. Another mechanism is intracellular targeting. Some amps are able to squeeze through the membrane and cause internal damage to the cell by binding straight onto the central dogma components, thus inhibiting DNA replication, RNA synthesis, and protein synthesis. The targeting of the central dogma causes the bacteria to die from the inside out. Amps can detect not only bacteria, but also fungi and viruses in similar ways, as seen on the screen.